Yeah, welcome and thank you for joining us for the first webinar of the National Early Detection and Rapid Response Framework webinar series. I'm Kaylin Clements. I'm a social scientist for the U.S. Geological Survey. I'm also the partner engagement coordinator and one of the co-chairs for the National Early Detection and Rapid Response Framework. This series is a collaboration between the Association for Fish and Wildlife Agencies, or AFWA, and the various agencies from the Department of the Interior involved in the National EDRR Framework. Carrie Wickstead, the Amphibian, Reptile, and Invasive Species Program Manager for AFWA, is working closely with us on this series and is co-facilitating the webinar with me. And our first presenter I'm going to hand it over to is Erin Malloy. Erin, please feel free to start getting your slides ready. Erin is a geneticist with the U.S. Fish and Wildlife Service Northeast Fisheries Center in Lamar, Pennsylvania. Erin has a broad background in molecule ecology. And hold on, you took over my screen, my bio for you. Okay. A broad background in molecule ecology and has worked for the service since 2015, building eDNA capacity. Erin is currently the lead geneticist working to integrate eDNA metabarcoding in the Great Lakes Basin-wide Aquatic Invasive Species Monitoring Program. And Erin, please take it away. Thanks.
All right. Thank you, Kaylin, for that introduction, and welcome everybody. So we're going to keep it pretty broad and high level today, looking at sort of our eDNA and an introduction to it. We'll start with just what is environmental DNA. So there's a lot of different definitions out there for what environmental DNA is and what they include in that definition. I like this one. It's pretty simple. It's pretty broad, but it defines environmental DNA as just a complex mixture of DNA from many different organisms found in an environmental sample, right? So really generic. But I think the important piece of that is that the DNA is from many different organisms, right? So if we envision this field crew here from our Green Bay Fish and Wildlife Conservation Office and the water sample that they're about to take, that water sample is going to have DNA in it from bacteria and fungi and viruses, algae, maybe the fish that are in that water, all the terrestrial inputs that are coming into those systems. So the mixture of DNA is incredibly complex that's in that sample. And then the environmental sample itself can also be very complex, right? So they're not all one and the same. We're taking samples from all across the landscape. It could be water or sediment or pollen or gut contents. And the material that we recover from those samples from the environment, it's going to have that complex mixture of organisms that release DNA into it, but it's also going to vary highly in its concentration and the quality of the DNA that you get out of it, right? So some samples are going to be really high concentration of DNA, high quality DNA, where others are going to be the opposite, right? They're going to be real low quality DNA, low concentrations of DNA. And those are all important factors that go into how we handle those samples, how we collect those samples, and how we process those in the lab, right? So it is a really complex sample type to work with. Now, the vast majority of eDNA sampling that's taken place right now is really with water samples. And so a lot of the presentation today will kind of focus around water samples. One, it's a common sample type, but it's primarily what we work with at the Northeast Fisheries Center. So one of the most common ways we collect water samples is through filtration. So this is just kind of giving you an idea regarding the equipment and the cost of equipment to get started collecting water samples. And it's quite variable. So on the low end, it might only cost you $150 to buy all the sampling equipment that you need, or upwards of $8,000 or more for fancy backpack units. What you're buying for all that additional money is efficiency at collecting samples and the convenience of collecting samples. The quality of the sample is the same. You can go out and do eDNA studies and collect quality samples with a hand pump and minimal input, right? So again, it's really efficiency and convenience that you're buying with that additional money. All right, so when we go out in the environment and we collect a water sample, what is it that we're actually collecting? So in this example, maybe we're targeting brook trout. We take a water sample, we're not actually capturing brook trout in this instance, right? We're only capturing what that brook trout's leaving behind for us to capture. And that can come from a lot of different sources. It could be free DNA that's released from metabolites from the brook trout. It could be skin and scales and mucus that are sloughing off the fish. It could be dead carcasses that are in that system from individuals that had passed away or predated. It could be during spawning, so you got gametes that are being released into the water column. But probably the largest contributor to eDNA that we detect is from the feces. So there's a lot of epithelial cells that are sloughed off in the process and released through defecation into the environment. So it's a mixture of sources that we're detecting in those samples. As far as a general eDNA workflow, it really starts with that collection component in the field. Here I'm showing, filtration taking place, but again, think back to all those different sample types. Depending on the sample substrate, that collection phase could look quite different. But here we're collecting water. That water can either be filtered on site, or you could grab a grab sample, and then it might go into a phase where we're doing some sort of filtration or centrifugation if you have a high turbid area that you can't filter enough water. And then it moves into laboratory processing. So that's the stage at which we're doing our DNA extraction, and we're using our molecular detection tools to generate the data in the lab. As far as laboratory processing and how we analyze samples, I'm going to talk about 3 different methods here. I want to concentrate on just the first 2 right now, the quantitative PCR and metabarcoding. We'll compare and contrast those, and then we'll talk about the field-based one in a minute. The main difference between the 2 is quantitative PCR is really target specific. We're only going to be looking at one species, or maybe a couple if we multiplex that assay, but it's really a limited number of species that we're getting information on using quantitative PCR. So we have to know what we're looking for. It's predetermined, it's built into our assay. So we have to know what the target species is. Metabarcoding is quite different in that we don't necessarily have to know what the target species is. We design the molecular assays in a more generic framework. It allows us to pick up a wide diversity of organisms, so we're doing more of a community-based analysis. They are typically directed in some way in that there's a taxonomic group that they're designed for. So in this instance, maybe we're looking for fish, or it could be benthic insects. It could be something like bacteria, right? Just a broad taxonomic group that we might be looking for. Quantitative PCR is based on fluorescence detection of the DNA in the samples, where metabarcoding is actually sequence-based. So we're getting the underlying data from the sequence of the DNA that was left behind in the environment. They're both quite sensitive assays, but I would say that the quantitative PCR is generally a little bit more sensitive than metabarcoding is for the target species that you're looking for. There is some differences in processing. qPCR is a little bit faster to process in the lab, maybe 2 to 3 days versus a week or 2 for metabarcoding. Metabarcoding has some additional steps that take place in the laboratory side. The collection in the field is essentially the same for either. That additional work is in the lab. We consider both of them to be high throughput in that we can process, hundreds or thousands of samples using either one of the molecular methods. The cost of them is relatively comparable. So the numbers that I have up there is an approximation, but the low end of that would be what it costs us to process a sample in the lab for all of the reagents and plastic consumables that we might use in the lab. The high end of that would include, personnel costs into that process. Okay, so that's the two basic analysis methods that are most commonly used with eDNA at the moment, and those are both based in the lab. There are field-based ones based on a couple of different molecular methods like LAMP and CRISPR, so the molecular underpinnings of them are kind of different, the way they work, but they too are limited to a number of species. It's predetermined what we're looking for. They're based on fluorescence, which is similar to qPCR. It's by far the least sensitive of the 3 methods, right? So the sensitivity is not nearly as good, but you can process things quite quickly. So you can, process that in the field on site and have information in 2 to 3 hours for processing that sample. The throughput on them is quite low though, so you might only be doing a handful of samples at a time. But the cost is much lower on it. So even though the sample throughput is low and the sensitivity is lower, field-based things still have their context in which they're useful, right? So absolute sensitivity isn't always the bar that we have to meet with eDNA. Sometimes sensitivity is not as important. Maybe that information up front in a quick and timely manner is more important, right? So like I said, those are kind of the three basic ways that we're analyzing eDNA at the moment. And they all have their strengths, they all have their weaknesses, and it really depends on what your question is and what your sampling design allows for. All right, so how do we use eDNA? So again, this is kind of in context of a water sample. We use it a lot for aquatic invasive species detection. That's probably the largest area. You can use it for endangered species detection. Anytime you're looking for something that's rare in the environment, it's a good choice. We do biodiversity or community assessment with eDNA. So this is that metabarcoding application. There's applications for fish passage, looking at below and above barriers to see if migratory fishes can pass and move upstream. And then I have distribution mapping that might be in a context of, say, like we do brook trout work where we might need to survey a couple of hundred tributaries and headwater streams to see if they contain or support brook trout. So we might go out and do a broad sweep with eDNA, get that information and use it to direct the field crew to do full-on assessments of those areas to characterize the distribution of that species in that context. But think again back to that slide we had of the different environmental sample types. Like if we were talking about a pollen sample or a fecal sample, this slide would look very different, right? So how we use DNA can very much be related to what that sample matrix is that we're sampling and what the target species are and the questions that we're asking. So yeah, this is kind of a broad overview of what it might look like from a water sample. Keeping with that theme of a water sample, what does that eDNA detection mean? And it really boils down to something really simple. It's the eDNA detection is confirming that the DNA of the target organism was in your sample. That's it. It doesn't tell us a lot of other things. It tells us presence, absence of DNA in that sample. It doesn't necessarily tell us about the status of that detection. It could be an alive animal. It could be a dead animal. Dead animals still shed a lot of DNA into the environment. It doesn't tell us anything about the origin of that sample, right? So animals move material around. So, it's an example of an eagle and, again, feces from that eagle will contain, fish DNA of whatever it was eating. And they, of course, move across watersheds and will move that DNA with it. People are really good about moving DNA around through boat traffic and other aspects of human activity on the landscape. eDNA detections don't tell us anything about the life stage. It's going to pick up eggs and larvae and fry or adults. It doesn't matter. They're all shedding DNA into the environment. That's all detectable. And an eDNA detection doesn't also tell us much about proximity, right? We get an eDNA detection. It doesn't mean that the organism is still in that area. They move around, the DNA moves around. So, we don't get a lot of power when we're talking about, proximity to that detection of where the organisms are. That said, it does give us a lot of information quickly. We can cover a lot of data points on the landscape, and even that presence absence information on a large scale is incredibly powerful information to have. All right, so what makes for successful eDNA projects? And I think that really comes down to communication, and it's communication before and it's communication after. That communication before piece is, talking to an eDNA lab before you start doing work, right? Speaking to the labs will help you clarify your question. It'll help you choose the appropriate sampling and analysis methods. Labs can help with the study design, where to sample when, how many samples to take, and they can help you with the budget and identifying funding for a project, right? So it's really important to have that upfront communication to set the foundations for a successful project. The communication piece after is also important, especially when we're talking about something like aquatic invasive species where there's a lot of sensitivities around the data itself. So it's important to identify the partners or the resource managers or the stakeholders that would be interested in that data, working collaboratively with them to develop data interpretation guidelines, having defined notification pathways and timelines for that information, defining what the data sharing expectations are going to be, and having an understanding of what your response options are based on those eDNA detections, right? So it's that communication, making sure that all of the various stakeholders are in the loop and informed and part of the process is, again, really important for successful eDNA projects. For eDNA resources, there's a lot of resources out there. There's a lot of web resources. There's a lot of information in the primary literature. I'm just going to highlight two things here. One is the Resource Manager's eDNA Toolbox. That's a product that's coming out of the National Framework. This is a USGS-led enterprise. So it's going to be a web resource that will have information about, eDNA, what is it, some of that introductory kind of information. It's going to talk about, the tools and how we can use them, what assays are available, what species we can do eDNA work with, and information about labs, how to contact labs, and the process and the pathways to taking on and networking and getting yourself involved in an eDNA process project. So that's a lot of information in there that will be coming online. I think that should be released to the public relatively soon. The other thing is training. So the U.S. Fish and Wildlife Service runs an in-person training session at the Conservation Training Center in West Virginia. So that's open to everyone. They can sign up and take the course. It's taught every other year. One year it's taught at NCTC. The next year it's taught at one of our U.S. Fish and Wildlife Service genetics labs around the country. So it does move around a bit. And there's also resources that go along with it. So we have an environmental eDNA sort of best management practice guide. It covers a lot with like project planning, study design, communication, data interpretation, those kind of aspects of it. That document's actually available online, and anybody can access it. We use it as a bit of a teaching resource for that course. Speak a little bit about emerging eDNA technologies. So there's a lot of buzz about eDNA. There's a lot of development and research done on eDNA, and sometimes eDNA seems like this magical thing that can do everything. And it's pretty exciting what it can do. And there's, like I said, tons of innovation. Some of that innovation is in like autonomous sampling and using drones. Some of that innovation is taking place in the labs with eDNA sensors or RNA or CRISPR or those molecular-based advancements. But the thing I want you to kind of take home about like all these exciting emerging technologies is that they all exist on a spectrum, right? So they exist on a spectrum that there's like sort of conceptual research and development applications versus what's monitoring and management applications. And they're not all the same. So you might read about something that's really cool in like a popular science article or in the primary literature, but it might be something that's firmly in that research and development phase. It's not necessarily something that we can use in monitoring and leverage that information for making management decisions. So it is important to keep it in context and where it exists on that spectrum. But undoubtedly, though, there's an incredible amount of stuff going on and a lot of really exciting things happening within the space of eDNA. The last thing I just want to mention is the Department of Interior Initiative, the National Framework Early Detection Rapid Response for Aquatic Invasive Species. So there's a lot of different components of this that support AIS work around the country. I just want to highlight, a couple of the things that are eDNA based, and the primary one is the Molecular Lab Network. So that'll be 3 U.S. Fish and Wildlife Service labs that'll be set up to handle increased processing of eDNA samples. There will also be a USGS lab that's there for science support to support the Molecular Lab Network. And that's really designed to build the capacity within the Department of Interior to work with partners and do more environmental DNA work on the landscape and further build this early detection and rapid response network for invasive species. All right, and that is everything I have. So thank you, everybody.
Thanks so much, Aaron, for that overview and for addressing many of the topics that people were requesting to learn more about. Our next two presenters are going to share about some on-the-ground work with eDNA and EDRR. First, we have Grace Lopp now. Grace is the invasive fish coordinator for the Minnesota Department of Natural Resources. She has worked on both sides of U.S. Fish and Wildlife Service's eDNA program, both as the federal biologist collecting samples and as a state partner using eDNA data to inform management decisions. And it looks like we've got your slides up, so take it away, Grace. Thanks.
Awesome, thank you. Yeah, I'll be talking to you a bit about the invasive carp eDNA program in Minnesota, and I'm also doing this presentation along with Pat DeHaan, who's the project leader at the Whitney Genetics Lab for the U.S. Fish and Wildlife Service, and his group handles the processing of the samples that we use for our program management decisions. So next slide, please. I'll give you a bit of an overview to start on what Minnesota has used eDNA for in the context of invasive fish. Minnesota DNR has been using eDNA to monitor for invasive carp since 2011. We've been working in partnership with Fish and Wildlife Service since 2014. The initial use was squarely for early detection. Our invasive carp in these locations that we're testing, that was unknown at that point. We had very sporadic captures of invasive carp in 2011. So this was helping just kind of determine where the carp were located in the state. Currently, we use eDNA to help monitor key locations where invasive carp are already known to be present for changes in the detection rate. This helps us respond to changes in the population and target locations for removal. So there's been a bit of a change in that use over time at the state. The Fish and Wildlife Service takes and processes the samples for eDNA. They use grab samples, essentially dipping water like in the photo here, and they're processed for bighead, silver, and general big-headed carp eDNA markers. We also get some eDNA data from USGS. Since 2021, they've been sampling some select sites using filtered samples and processing them at their laboratories using their own markers. So next slide, please. So how are we using these data? These data help us monitor for changes in eDNA detection rates by season and year. We're looking at some key locations where we frequently capture invasive carp. And we're also looking at some kind of key locations where we expect invasive carp might stack up. Places that act as barriers to movement where we could see aggregations of invasive carp. Excuse me, sorry. I have a bit of a cold today. But these data help us inform what time of year and what locations we should be targeting for removal efforts. And it also helps us add a line of evidence for changes in abundance of invasive carp in the state. We also use capture data from fish that we've actually captured, telemetry from tagged invasive carp, and data from sightings of jumping invasive carp to help us identify locations to sample and also get kind of a macro level scale look at what changes in abundance might be occurring in Minnesota. Having these different lines of evidence is important to us because we do not have capture data to support modeling the abundance of invasive carp in the state yet. So we use a variety of data to help us sort of get a picture of whether invasive carp are increasing or decreasing in abundance in the state. We're currently sampling locations in Mississippi River Pool 8, which has been a large portion of the invasive carp captured to date have been from that location. Pool 5 and as well as Pool 5A. Those are locations surrounding Block and Dam 5, which is a major barrier to fish movement in our state. We also have Fish and Wildlife Service sample the St. Croix River and the Minnesota River to help us see if abundance might be increasing or decreasing in these areas that are sort of at the extent of where invasive carp can move in the state. Next slide, please. There are some concerns regarding eDNA that I wanted to make sure folks are aware of. When we first started sampling for eDNA back in 2011, we were working with a university partner on this project, and the samples revealed some surprising results. We believe that there were false positives that occurred during that sampling, suggesting a much higher abundance of invasive carp than were present at that time. So that kind of created some distrust maybe at our agency for eDNA. But since then, we've sort of, again, switched our focus from early detection to monitoring. I think that helped build some trust. Also working with these federal agencies that have developed very strict protocols to prevent contamination has helped build trust. And having genetic markers that are very selective and only show results for the species that you're trying to detect, that's also been important to helping kind of build that trust back. There are challenges in interpreting data from multiple sources. I talked about how we use sample data from Fish and Wildlife Service grab samples that are centrifuged and processed according to their protocols, as well as filtered samples from USGS that are processed according to their protocols. So we do get differences in detection rate from the two different agencies, and that can create some challenges in knowing how to interpret those data. There's also always a challenge in communicating with the public what eDNA results mean. I think Aaron did a good job covering what eDNA can tell you and can't tell you, and just it's important to make sure that those things are communicated out to the public so they know how to interpret the results for themselves as well. Next slide, please. So our process starts with planning in the beginning of the year. We meet with the Fish and Wildlife Service eDNA coordinator, Nick Fronauer, who's on the call as well, to help plan next year's sampling sites. So the sites that Fish and Wildlife Service samples are in response to state partners' requests and needs. The Fish and Wildlife Service field team then communicates with us when they will be out conducting the sampling. This helps make sure that our own crew is not causing a disturbance or introducing some sort of confounding factor that might influence their results. It also helps make sure that we can provide them with up-to-date data on the river conditions so that they're appropriate for the eDNA sampling to occur. And they sample during both spring and fall. Next. And then after the samples are processed, we get the results from the Fish and Wildlife Service. They're sent to both Minnesota and Wisconsin because most of these are border water sites, so that both receive the data at the same time. The data are provided to us in a spreadsheet format and also in a PDF that contains maps of the detection results. I have an example of one of the maps here. You can see it shows where all the samples were taken at that particular site, and the different colored shapes show where positive samples were located for silver carp, for the generic marker for both big head and silver carp, and for the big head marker. The states are given an opportunity to make a press release prior to Fish and Wildlife Service posting those results publicly. We consider a press release if the results indicate a new development, such as a range expansion or large increase in abundance. Otherwise, it's just kind of another part of our monitoring program, and we include information about the results in our annual reporting. And then Fish and Wildlife Service posts the results on a public-facing dashboard, which is available in the Wakelet as well. Next, please. So key lessons we've learned along the way: it's key to have sampling Key to have communication, both pre and post sampling among agencies to make sure that we are getting the best possible results, that we're getting the information we need, and that the Fish and Wildlife Service is also aware of how to conduct this sampling, because we might have some on the ground knowledge we can share with them. It's important to know about the species you're sampling for and how their biology and ecology might factor into when to sample and where to sample. We also want to make sure that there's strong protocols in place to keep quality assurance and quality control high. We're not looking for a situation where we have false positives that might skew things, or that we're not detecting eDNA when it's there. So having those strong protocols is very important to building trust and to be able to interpret the data. eDNA can be useful for early detection, but it's also useful for more than early detection. It can be used as a monitoring tool as well, and that's sort of where we've shifted our program. And this partnership has created some additional research and monitoring opportunities. We're looking to see how we can relate these data to other data that we have, including the capture data. And we've also used some eDNA for identifying larval fish and other things like that. So it can be useful for more than just the already proven things. And it's good to have those research opportunities as well. Next, please. So the funding is provided by the Fish and Wildlife Service's Aquatic Invasive Species Program base funds for sample collection, laboratory analysis, and results reporting. Their lab processes roughly 9,000 to 10,000 samples per year for their entire project. This covers both the Great Lakes and the Upper Mississippi River Basin. And they also have some capacity for research and methods improvements. And so if you have any further questions on funding and capacity from the Fish and Wildlife Service end, I'd encourage you to reach out to Pat. Next, please. So thank you for the opportunity to talk with you all today. If you have any questions, please put them in the chat, or feel free to reach out to me or Pat directly. Thank you.
Thanks so much, Grace. Thanks for sharing about your agency's experiences. Our next presenter is Christine Dunker. Christine is the South Central Alaska Region Invasive Species Program Coordinator with the Alaska Department of Fish and Game, Division of Sportfish in Anchorage, Alaska. Christina spent almost 2 decades working on aquatic invasive species in Alaska, and will be telling us about her work with eDNA and her program.
Great, thank you for that introduction. And I'd also like to acknowledge my co-author, Adam Sepulveda with USGS, who has been an incredible partner on my program's eDNA work. Like was mentioned, I'm going to be presenting a very brief overview of Alaska's use of eDNA detections of invasive northern pike. Please advance. Okay, and I want to start with just a very brief context, super quick, about my program's work being mainly focused on this species. And with northern pike, our efforts are on eradication, suppression, and research. All of this is in South Central Alaska. Northern pike are a native species in much of the state of Alaska, and where they're invasive is only really south of the Alaska Mountain Range, which is in the red solid areas on this map. In our current situation, it has been discovered that northern pike in this area are having very significant impacts on salmon resources. If you could please advance. And over the last 2 decades, please advance again, we've had a dedicated program in place to contain their distribution and manage their impacts and their abundance. Next slide, please. In 2014, our program began to incorporate eDNA into our work. Please advance. Our initial foray into eDNA was a collaborative effort with the Fish and Wildlife Service Conservation Genetics Lab in Anchorage to develop the first eDNA marker for northern pike and to ground truth its effectiveness with Alaska populations. Around this time, we were amid our largest northern pike eradication project, where we were removing northern pike populations from an entire drainage on Alaska's Kenai Peninsula. We collaborated with Adam, as I mentioned earlier, and Ora Russ with Fish and Wildlife Service to study the efficacy of using eDNA to evaluate rote-known treatment success. So this study involved investigating detection probability with distance from caged pike. And we also looked at detection timing after the caged pike were killed, as well as post-treatment detection in 4 recently eradicated lakes. So in our case, our study found decreasing probability of detection with distance from that source of northern pike. But our biggest takeaway was that eDNA really didn't serve our purpose as an eradication evaluation tool because of latent DNA within water bodies being detectable at least 8 months following treatments. And in most cases, our treatment evaluations needed to happen much sooner than that. Please advance. And then in 2018, we teamed up with the USGS again to test portable field-based DNA platforms for potential application in our work. In this case, we found many of our results to be affected by inhibition, and with that, we concluded that traditional sampling and lab-based PCR was most efficient for our needs. Next slide. So since these initial studies, eDNA has been a part of our toolbox for early detection monitoring for new northern pike introductions and to help assess if water bodies are free of northern pike before we invest significant resources into prevention or eradication in connected waters. Please advance. In 2022, we developed a pretty extensive adaptive management plan for invasive northern pike in Alaska. This plan also includes a great deal of technical guidance and SOPs, advance, of which we have an SOP dedicated to eDNA data collections. Within this SOP, we have a model, please advance, that was developed from our 2016 study that helps us gauge detection probability given different variables of population size, littoral acreage of a lake, and the number of 1 liter water samples collected. This model is for lakes only. We don't have a similar tool yet for flowing waters, which is something we're hoping to develop in the near future. Next slide, please. While it is a heavily relied upon tool here in Alaska for invasive northern pike detections, eDNA has not really been a one-size-fits-all method for us. And we've had to adapt our responses to positive eDNA results to the unique characteristics and risk tolerance for each situation. Perhaps our biggest challenge is with distinguishing if the DNA that we collect is from a live pike or another source that introduced pike DNA, as we heard about from both Aaron and Grace. For example, in at least 2 lakes in Alaska, we believe we were detecting pike DNA that was trapped in disturbed sediment rather than from an actual fish. So interpretation of results, especially in cases where we might only have 1 positive, is a huge challenge where, as managers, we need to make judgment calls based on the risk of invasion versus the risk of being wrong in our interpretation of results. We also struggle with how to prove the negative. Not detecting DNA doesn't necessarily mean that pike are absent from a site. While we have tried to address the probability of detection in lake systems through the model I just mentioned, it's not a perfect system, nor one that assists us in flowing waters presently. So we're still working to develop stronger sampling protocols. And the other major challenge is the timing it sometimes takes to get our results back. Like all programs, ours is resource and funding limited. We're super fortunate to have wonderful partnerships with labs that process our samples for virtually no cost, but understandably, our samples are then not always high priorities for processing. Sometimes it can be many months to receive results, and within that time, the situation in the field may have changed. Lastly, as we also heard in Aaron's presentation, technology keeps changing. Almost as soon as we make investments in new equipment. Our eDNA SOP, which is only a few years old, was written mainly from taking discrete grab samples and filtering in the lab with peristaltic pumps. So with backpacks and citizen science samplers and autonomous samplers and we have new investments to make, and I suspect that that'll keep continuing as technology advances. Next slide. Despite some of the challenges we work through with eDNA, our program is in a really strong place with communications of results getting to the right people quickly. Positive eDNA results are much like other invasive species reports in our program's case, and we have a standard communication flow, which is outlined in our pike management plan for how that information gets disseminated. Next slide, please. And so to wrap up, my team has been really fortunate to have been some of the first trailblazers of eDNA work in Alaska. Over the last decade of our involvement, we've learned many lessons and are continuing to learn more. For example, with the really technical details, we've found there is a definite time trade-off in choosing which pore size of filter to use. And we continue to debate if we're better off filtering less water through finer filters or more water through porous ones. We've yet to finalize a decision there. But I think we're coming to understand that results interpretation is our largest challenge and can be more of an art than a science, or at least a good exercise in logic. But sometimes positive results can generate more questions than answers and can trigger some pretty difficult management decisions. So going in with a clear understanding of our risk tolerance is important. And it's totally fine for that risk tolerance to be different based on unique circumstances of what we're working within. And we continue to learn to adapt with technology. Most of our program's efforts are funded through grants. So we've learned to increase our eDNA capacity by building funds for equipment and what's needed into our grant applications so that we can purchase new equipment and process samples as timely as possible. And with that, it's very important to build strong partnerships with open and clear communication. Successfully building eDNA into our program has only been possible because of these strong partnerships we have with Adams Lab, with USGS, and ORAS here in Fish and Wildlife Service in Anchorage. And we're extremely appreciative of those partnerships. Last slide. And I think we are probably out of time for questions, but my email is here if anybody has any follow-up questions for my program's use of eDNA. Thank you.
All right, thank you so much to our presenters. Some fantastic information that has been relayed so far. So we are going to build a space for some questions before we jump into our breakout rooms, where you'll get a chance to provide some feedback on some of your experiences that you might have had with eDNA. So I'll encourage you to continue posting your questions in the chat. And then also, if you'd rather raise your hand and ask a question, then you can do so. And presenters, this is going to be on you as well as the eDNA team. So feel free to jump in if you can answer the question. So the first question I have is from Brian Engelbert. And when talking about field-based analysis methods like LAMP and CRISPR, is the less sensitivity more likely to result in more false positives, false negatives, or both? So this might be a question for Aaron, or I think Stephen is on the chat as well.
Yeah, the sensitivity would lead to more false negative results, right? So you would be missing something that was there. It wouldn't necessarily detect something that wasn't there. It's missing the things that might still be in the system. That said, again, just remember that even though those technologies are less sensitive, they have their application points. So, it might be a port of entry where you're looking at a shipment of ornamental fish for an aquarium trade and you want to screen it. Those things are all, compact or together. Like you're going to get the gobs of DNA out of that kind of sample, right? So sensitivity might not be the ultimate goal there. It's the quick determination so that the shipment can move on. The same for maybe in a hatchery if you're screening for pathogens from water samples in hatchery where the pathogens might be in relatively high concentration. So, there's use case scenarios where that field-based detection makes a lot of sense, even though they're less sensitive.
Great, thank you. And the next question is from Michelle Tremblay, and she wants to know, is there a central library of species for AIS detection, even though species native to one North American region but could be found in another? And if so, how can we contribute to it? And will our data collection be vetted with our own metadata?
I can jump in again if nobody wants to take that. So yeah, a lot of the genetic resources, if they're deposited in public resources like NCBI or GenBank, everybody that's creating those reference data sets are harvesting information from there. So, you know, that makes it into everybody's data sets. And references from outside the region of study are still useful in that case, but it's always best to have references from the region you're studying, right? But like we would include everything regardless of region. If that answers everything there, I don't know.
Thank you, Aaron. If anybody else wants to, on the team, would like to jump in as well, feel free to.
Hi, I'm Cheryl Morrison, and we are involved with a genetic material repository for invasive species. So kind of trying to set a high bar for the quality of the samples that are vouchered, and all the information about the sample collection will be searchable on a database and can be requested for assay development. So that's one step towards a kind of a group effort to have some samples available.
Thank you, Cheryl.
Sure.
Okay. And then another question that came in earlier, and it was from Caitlin Parrington, and she wants to know, how effective is eDNA detection on larger lakes for EDRR, specifically when it comes to detecting fish? And I know that part of this was answered, but if anybody wants to jump in with a little bit more information, that might be helpful.
I'll jump in again here. Yeah, so a lot of the work that we do is actually on the Great Lakes. So, I mean, they're relatively large, I guess. So what we're seeing, at least, and that's all metabarcoding work, so which is a little less sensitive than qPCR. What we see is that it's considerably more sensitive than the traditional gear efforts that we have on the landscape out there. And, you know, we have pretty extensive traditional gear where we're using, you know, multiple different types of gear sets, and even all that data combined, we're still seeing higher sensitivity and higher detection rates with eDNA work.
Thank you. And Adam has also shared a paper in the chat, and we'll make sure that we add that to the Wakelet as well, if it's not already in there. So now what we're going to do is we're going to jump into some breakout rooms, and we're going to get some feedback from you all. We have 4 main questions that we'd like to have answered, and some of it relates to your experiences with eDNA, as well as any regulatory barriers that you might face with implementing eDNA. And, you know, based on some of the information today, what you've learned, what caught your attention, what might you want more information about, you know, what could you use? And this is going to help us develop these future webinars in this series, and also resources and products. So I'm going to open breakout rooms. You should see a notification to go to a breakout room. Forgot to record that, but I wrote notes, Stephen, so.
All right. Well, in room number 2, most of our folks had more limited experience using eDNA. You know, we spent a lot of time talking about the barriers, and the main barrier that came up with our group is just limited knowledge. And so folks are really glad to get more information from the 3 presenters today. Another barrier that came up that sounds like it was common in Steve's group was just capacity of labs to process samples, and the ability of labs to turn results around in time to make meaningful management decisions. We also talked a little bit about standardization issues, and just making sure that, results were consistent across labs, and that data was reliable. We did have kind of an interesting side conversation about citizen scientists and the value of citizen scientists in eDNA. We talked about some of the pros of that approach being the ability to get a lot of people out on the landscape, and the ability to really engage people and get them concerned about, the species that are being monitored, whether they're T&E species or aquatic invasives. And some of the cons we talked about with citizen scientists was just, making sure that the quality control is there, making sure that consistent protocols are applied. But we did have an interesting conversation about, how we might engage citizen scientists in eDNA efforts.
Awesome. Thank you. Okay. Group number 3, Cheryl and Nicole.
Hello, everybody. So in group 3, we got through all the questions. The barriers, one of them were eDNA is a new technology, and it's hard to design good experiments and interpret the results well to communicate to the public and managers. So help with that design and communication and interpretation is definitely needed. Cost, how to design a study to cover a very large area in a cost-effective manner, and the expertise to do the assays themselves. So those are all things that were potential barriers. When it comes to good experiences, there were a few studies that had gone really well, but there were invasive species detected. In one instance, they were able to quickly take some eDNA samples when they unexpectedly caught invasive species fish, and they were able to learn about the spread of the eDNA through the experiment. Another one involved more extensive sampling, and the eDNA data made sense as far as where the species was detected along I think it was a lake. For problematic experiences, again, transferring a specific eDNA sample to a larger area, is that possible? How would you do that? And then many people said that the expectations of what the eDNA results and how to communicate them is difficult, because people have some expectations that this is just the magic result, but the results are often nuanced. And so how do you interpret that and explain? And then for just first-time users of eDNA, the methods can be challenging. You go out and you try something and it doesn't work. So then what do? You have to go back to the drawing board. So maybe having some more expertise in study design so that you can avoid some of those challenges. And then everybody seemed very excited about the real life examples that were provided by the speakers. They felt that they were great to see how, for example, how the Alaska pike, how that immediately those results were rolled into management plans. And yeah, and I think that's about it.
Awesome. Thank you.
Sure.
All right. We're going to roll to group 4.
Good afternoon.
Yeah, go
Thank
ahead.
you. Yeah. So we had a good discussion as well in our group. Sounds like the breakout groups were really productive. For some of the barriers, some of the things that hadn't come up yet that we talked about were how to deal with inhibitors, especially based on where sample collection may occur and what the environmental conditions are. And so the limitations of applying eDNA in those situations was definitely a barrier. Another barrier was understanding about what the results meant related to response options. So kind of coming up with having more conversations between the labs and the managers about what the results interpretations meant, and then working with the managers on how those response options could be focused. Another barrier mentioned was some of the folks were dealing with a really focused target list of targeted species, but others were dealing with a really broad array of potential targets, ranging from amphibians to mussels to other aquatic organisms as well. So not sure if there were markers available for all of those targeted species. Some of the good experiences folks had were really rapid responses for the labs that they needed, that they were working with, and getting results quickly, which had really important impacts to be able to assess potential invasive species and especially with the new potential invasions. So being able to respond quickly was really important to them, and they really appreciated that. Some of the problematic areas really, in some ways, have improved, or that number of problematic areas has reduced over time, especially as the technology has improved for sampling. So it's a lot easier to get out and collect samples with much limited concerns about contamination with some of the filtration systems that are available. Folks really liked that. Another problematic area, though, was finding the funding to actually process samples. That's a challenge. Folks mentioned things that they highlighted or heard today, such as some of the automated and passive samplers were of interest, as well as some of the meta barcoding and broader applications beyond just qPCR.
Okay, great, thank you. So we've got 6 more groups and 4 minutes, and I know some people have to jump. So we do have your information, all of it written down. Were there any like big standout things from the remaining groups that haven't been covered yet? From groups 5 through 10. Anything that would be better for the larger group to use? Go ahead, Jenny.
Sorry, I don't know how to raise my hand on Zoom. Really quickly, one of the really positive things was that this tool using eDNA is a really great one that fosters partnerships and allows groups to work together between different agencies and organizations. And so that it's a really positive experience working with eDNA and helping us collaborate.
Thank you. Katie?
Yeah, I think a really positive experience was someone in our group was using eDNA and got positive detections for a species that was thought to be extirpated, and they were able to go back out and find it. And so being able to couple these broad eDNA surveys with to help you focus on a particular area was found to be really useful. And then just as terms of barriers or difficulties is the communication, especially with invasive species detection. How do you communicate that without scaring people?
Yeah, thank you. David?
Yeah, we were just getting into a lot of the topics that were covered, we discussed as well, but one thing we were getting into at the end there was this idea of latent DNA in systems, as well as just allochthonous inputs of DNA that cause essentially false positives, and the best way to communicate that with the public and with managers, because it drives uncertainty and skepticism.
Definitely. And I see in the chat too, Devin mentioned the concern with false positives and false negatives, potential costs associated with making those decisions based on those results too. So, all right. Well, I'm really excited to dig through all the information. I'll have to say I was creeping on all of the different Word documents and watching them get populated. So it's going to be really nice to sit down and go through all of this, and we appreciate your time. And we really appreciate all of our presenters and folks who've been helping facilitate and note take and everything. I also want to give a big shout out to Kaylin, who's been pulling us all together and coordinating in multiple fronts and sending out so many emails and everything.